All right. This is a little bonus thing that I want to do because we just talked about hemoglobin in biochemistry class. Hemoglobin lets us understand how diseases work on the level of cells and below cells when the hemoglobin is broken. And we have a case where there's a disease where we can even have some therapies that come about because we understand the biochemistry of hemoglobin. The disease is sickle-cell anemia. And the first time that the disease was narrowed down to a problem with particular blood cells was when microscopy was applied to the sample of the blood of a dental student from Granada. Now, the Chicago doctor himself did not have this disease, and this disease was not looked into very early on because it's not prevalent except among several populations from Africa. The Granada dental student provided the first example that we have where we see something is really wrong with the red blood cells here. So what's going on and how can we fix it? This is the red blood cells. Even to somebody like me who doesn't usually look at cells, it's very clear that there's something bad going on with these red blood cells. They look like little sickles when they should look like little donuts. So the way the disease works, it turns out that you can actually be heterozygous for the disease and never develop sickle cell anemia. You can have a particular gene, and only if you have 2 copies of that gene will you develop sickle cell. Somebody heterozygous will actually be protected from malaria, and that's why sickle cell has survived and been passed on in regions where malaria is endemic. 1 in 12 Africans, as an example, are heterozygous. And so you can see that you have this big structural difference in the red blood cells. Not all the cells, but just occasional ones. It's enough to cause the disease. So how do we find those genes? And if we look at the whole human population, how many different genes do we find? And we trace it to a hemoglobin gene. So there's a mutation in the gene for hemoglobin that causes sickle cell, especially when it's homozygous. DNA sequencing, we've found 300 variants or more of the hemoglobin gene that appear to be important. These are called alleles. And remember that you get 1 allele from each parent. And so if you have 2 of the same allele, you're homozygous. And it's only if you have 2 of a very particular mutation that you get the most prevalent form of sickle cell. So the results, and of course the disease has been around much longer than microscopes have, the results are that sickle cell anemia shows up in African populations predominantly, and it causes weakness, dizziness, shortness of breath. The hemoglobin content of blood is half normal, so it's like an anemic person on the graph that we showed in biochemistry. This is because the sickle-shaped cells are fragile and they break apart, and so you have lower iron concentrations in the blood. The sickle-shaped cells also will block capillaries. This will cause pain and loss of organ function. That's me describing what it feels like, but it's more important to listen to the patients here. The patients, when they tell you about it, tell you that it's a lot of pain, unbelievable pain, jackhammer on your back, throughout your whole body. Sometimes in the joints, abdomen, in the head, in the chest. Basically, wherever there's capillaries, the capillaries can be blocked by an episode of the sickle cell blocking the capillaries. I wish people in the medical field would take the disease more seriously. I've had experiences where they've called me a drug seeker. Because it's happening in the capillaries, there's no big physical manifestation of it. You have to see that the patient, there's sometimes little things like jaundice, there's little things that you can see, but they're not the big kind of things, they're not proportionate to the pain. And so the 38-year-old, he says that he's just tired of the whole situation. Then we have an 8-year-old who says, every time I have a pain episode, I either get a heating pad, they take my temperature, a fever means you have to go to the hospital, and of course the mother knows exactly when this is happening because she can see the small kind of parts where the eyes start to turn yellow, someone is squeezing legs and arms, the pressure is actually coming from within the capillaries, not from outside the capillaries, but it feels the same way. So this is a serious disease. How can we get to the bottom of what's going on? If it has a genetic part, maybe we can figure out what that is and have a drug to physically block whatever's going on. And so the gene for hemoglobin sickle cell was labeled hemoglobin S. The non-disease gene is hemoglobin A. There's lots of other hemoglobins that have been found, gene variants that have some kind of disease. Hemoglobin S, we narrowed it down to what exactly in the gene causes the disease, and we got to a very small, a single nucleotide change, which changes a single amino acid. So hemoglobin S is where you have a glutamate change to a valine. And those of us who've memorized our amino acids, right class, have noticed that this is a very small change. These amino acids are chemically different, but it doesn't seem like this kind of change should cause this big of a disease. By the way that we show this is we say beta for beta chain, E6V is how we say that this mutation has happened. And in fact, let me show you right here. This is my little model that I've shown my biochem students. This is the beta chain of hemoglobin. And you can see there's the heme right here. Here's the two histidines that we talked about that sandwich the heme. And this also has the mutation for sickle cell. But to show it to you, I have to turn it around. There is the little glutamate, hemoglobin A, and hemoglobin S. This little thing is changed to something slightly smaller and without the charge on it. That one little change on the hemoglobin molecule causes sickle cell disease. This actually gives me a little bit of hope because it says one little change that has big effects, maybe we can make a drug, a little drug that goes after the little change. So why does that little change cause such a big change? Well, hemoglobin A and hemoglobin S, they form tetramers just fine, but the hemoglobin S has this pocket up here that because the charge has been taken away, it's become hydrophobic and therefore sticky. And because hemoglobin is a tetramer with two beta chains, you got a sticky patch on the top and bottom. That means in a place like a red blood cell where there's a bunch of these hemoglobins around, the sticky patches will start to stick together and you'll form a line, not just a glob. They start to line up, they start to stick together, they form a strand, they form a rope, they form a fiber. They literally are crystallizing in sort of one dimension inside the cell. And those fibers result in the weird spiky appearance of these cells. This is all from a single amino acid mutation. Because capillaries are so small, here's a picture of some capillaries. That's really small. Red blood cells sometimes are squeezing through. The fact that they get through is pretty amazing. And the sickle cell will stick into it. And so that's why you get the issues where you have the organs that filter the blood, especially. Kidneys will have lots of problems. They'll have necrosis from being blocked because there's lots of blood passing through. Liver will also have problems sometimes, although that's actually not shown here. It'll cause problems in the brain where there's lots of blood flowing through, and that's why you'll have the pain all over. So this is what sickle cell disease, it comes from a very small beginning, but it goes all over. So the red blood cells have very different shapes, and they even react differently to physical conditions. They even sound different. So they have this kind of photoacoustics. It's not really a microscope. It's more of a microscope plus a sonifier. And they actually will send sound waves in and see how the cells vibrate under under those. And they tested it by using sickle cells. And they saw that the red blood sickle cells will actually vibrate differently. And so it's like they make a different noise if you put sound waves in, or you use the laser to produce sound waves, I should say. So you use the laser to hit them like a drum, and the drum will sound very different. This just shows how different sickle cells are from regular red blood cells. So because they're so different, we can make simple diagnostics that can go pretty much anywhere on the globe. One of the diagnostics is using the fact that sickle cells will sink to the bottom of the tube. They'll be dense and fragile and things like that. So they actually put them in a little tiny tube and they spin them. And there's easy ways to spin something, egg beater, or you can just, it's pretty easy to get a centrifuge. You can even hand power certain kinds of centrifuges. So you can take this to places where there isn't even electricity, put whole blood into the tube. The tube has a gel in it that filters out the red blood cells, and the dense red blood cells will end up on the bottom. A person with more dense red blood cells is very likely for those to be sickle cells. So I just want you to see, and here's an idea. SCA negative doesn't have a band down there. SCA positive, you see a little red band at the bottom of the tube. Lots of dense cells. And so this means that these symptoms come up. All these symptoms that we talked about happen all because of a single point mutation. So like I said, could a small molecule block this? Well, there's lots of hurdles that molecule has to leap, but there is actually a drug going on the market, I believe. It's gotten some kind of emergency authorization. So I want biochemists to read about this. It's called vowelotor, and you can see it's a small drug, but I think it has a hope of actually doing something, because a small drug could possibly block polymerization. How does it know to get to the right place? How does it do it effectively? I'm really not sure, but that's why we have clinical trials. So the FDA has gone through those clinical trials. I want you to read about that, biochemists. But there's also skepticism, because how could a little drug be having that big of an effect? So read about it. Here's where the babies are born worldwide with sickle cell disease. And you have the, you can see that Africa has a large prevalence of this disease. And if you can map it out to the allele, here's the sickle cell allele. It maps out to Africa in the central region, especially sub-Saharan. You put that next to a map of malaria endemicity, and you see that they really match. So this is probably a pretty good explanation, because malaria is somehow messed up by the sickle cell mutation, especially when it's only heterozygous. And that was a question when I first started teaching this. Now I think it's pretty much been proved, and I want to show you the papers. Here's the paper that, to me, proved it. It showed sickle cell people are less infected, less affected by malaria. Why? Well, the fibers look like they interfere with the malaria parasite. It has a very complex life cycle, and it takes part inside the blood cells. So all these fibers will mess up with its life cycle enough so that it can't really replicate or get a hold on a person who is heterozygous for it. So it's not a feature, it's not a bug, it's a feature. Because in a sense, it's a feature to promote a disease to keep you from getting a worse disease. That's what sickle cell is. So here's the papers if you want more detail about how they know this. There's even evidence that selection for this trait is still happening, in the sense that the sickle cell allele is favored in the parts of Africa that have malaria. This is another kind of evidence showing that it's important to fighting off malaria, but now we can fight off both, right? And so here's a medical facility, and this is in Democratic Republic of the Congo. And if you can read the French, which is really close to English in this case, you can find sickle cell on the list of things that they offer treatment for. So how can you cure it? Well, the old treatment is hydroxyurea, because it would stimulate the production of fetal hemoglobin. And think about why that would help, biochemists. I've given you the clues. Fetal hemoglobin is different than regular hemoglobin. But now we've got CRISPR, for example, Nobel Prize this year, and we've got other therapies like the drug I told you about. Biochemists, that's where I want you to have the readings. So the way CRISPR can do it, CRISPR might be able to literally erase the gene. It's if any gene could be erased, I think this one would be high on the list, because it's a very small change. If we could erase that very small change, and we could give a person with sickle cell normal blood, then we would cure the disease. I want to mention Francis Collins, the head of the NIH. He also authored the book Language of God. I'm a sort of participant in BioLogos, which is an organization he started. He gave a really great lecture in 2017 that highlighted SCA for about 5 minutes. I used to assign this year, biochemist, don't worry, I'm not assigning it, but I do want to say, I think you should watch this, because he's got a lot of good stuff to say, even beyond the sickle cell angle. But that is not assigned. What is assigned is the 4 news readings, and this is our homework for the homework problems. Now I'm going to go and post this, activate the reading quiz, do all that fun stuff, and so thank you for listening. Biochemists, I will see you in class very soon.